William Coley was a bone surgeon at one of the most prestigious hospitals in America, New York Cancer Hospital, which would later become Memorial Sloan Kettering. For 40 years, he injected bacterial toxins into his patients' tumors and documented what happened. Tumors regressed. Patients with inoperable sarcomas survived years beyond any prognosis. He published his results. His colleagues could read them, and then the head of pathology at his own hospital decided that radiation was the future of cancer treatment. Not because radiation had better results, because radiation could be controlled by institutions. Coley's toxins could be made by anyone. I found this while researching how the FDA classifies biological treatments. I was tracing a regulatory change from 1962 that reclassified certain therapies as new drugs, trying to understand why specific treatments vanished from American medicine in a single decade. One of those treatments kept appearing in every source I read. A bacterial preparation used continuously from 1891 to the early 1960s, published in major medical journals, documented across hundreds of cases, and then gone, not disproven, not replaced by something better, reclassified into non-existence. The deeper I went into William Coley's records, the more I realized this was not a story about outdated science. This was a story about what happens when a treatment that works cannot be owned. It started with a death that haunted him. In 1890, William Coley was a young surgeon, freshly appointed to the New York Cancer Hospital. One of his first patients was a 17-year-old girl named Elizabeth Daschle. She had a sarcoma in her right arm. Coley amputated. The cancer had already spread. She was dead within weeks. Verify exact timeline of Daschle case. Some sources say 10 weeks, others say months. Coley was shaken. He began searching hospital records for anything that might offer an alternative to surgery. And he found something that did not make sense. A patient named Fred Stein, a German immigrant, had been admitted years earlier with an inoperable sarcoma on his neck. The tumor had been declared terminal, but then Stein contracted a severe bacterial infection, erysipelas, a streptococcal skin infection that produced high fevers. As the infection raged, the tumor shrank. When the fever broke, the tumor was gone. Stein was discharged. He was alive. Coley tracked Stein down. He was still alive years later with no recurrence. Verify exact year. Coley located Stein and confirmed his survival. But here is what the standard history tends to skip over. Coley did not simply note this as a curiosity. He went back through the medical literature and found over 40 documented cases of cancer patients whose tumors had regressed following acute bacterial infections. This was not one case. This was a pattern. Infections triggered something in the body that attacked the cancer. Coley did not know the mechanism. Immunology as a field barely existed, but he had the clinical evidence and he decided to act on it. In 1891, he deliberately injected streptococcal bacteria into a cancer patient named Thomas Zola, who had an inoperable sarcoma. The tumor shrank. Coley refined his approach. He developed a mixture of killed bacteria, Streptococcus pyogenes, and Serratia marcescens, that became known as Coley's toxins. The preparation induced high fevers. The fevers activated what we now understand to be the immune system's response to cancer cells. Over the next 40 years, Coley treated over 1,000 patients. He published his results in the Journal of the American Medical Association, the American Journal of Surgery, and numerous other peer-reviewed publications. His results were not hidden. They were not secret. They were printed in the most respected medical journals in the country for anyone to read. I spent months trying to understand why a treatment with 40 years of published results simply vanished from American medicine. And the answer has a name. James Ewing. Ewing was the head of pathology at the same hospital where Coley practiced. He was brilliant, ambitious, and he had a conviction. He believed radiation was the future of cancer treatment, not immunology, not bacterial preparations, radiation. Ewing had powerful supporters. In particular, he had the backing of James Douglas, a mining magnate who had made a fortune in radium extraction. Douglas donated heavily to the hospital. He funded a radium research department. He gave Ewing institutional power that Coley could never match. But what bothered me more was the specificity of what Ewing did. Ewing did not simply advocate for radiation alongside Coley's approach. He actively suppressed Coley's work within the hospital. He restricted Coley's access to beds. He criticized the toxin therapy in internal meetings. He used his position as pathologist to challenge Coley's case reports. By the 1930s, Ewing's influence had effectively marginalized Coley within the institution he had helped build. Radiation became the orthodoxy at Memorial Hospital. Immunology became a fringe interest. And here is the detail that makes this personal. Ewing did not have evidence that radiation produced better long-term outcomes than Coley's toxins for the cancers Coley treated. What he had was institutional control. Radiation required expensive equipment. It required specialized facilities. It required hospitals. Coley's toxins required a vial and a syringe. One approach concentrated power in institutions. The other distributed it to any physician who could read a published formula. Consider what that meant for the economics of cancer treatment. Coley died in 1936. His toxins did not die with him. Other physicians continued using them. His daughter, Helen Coley Knotts, spent decades compiling her father's case records. She documented over 900 cases. Verify? Exact number. Some sources say 896. Others round to over 1,000. She founded the Cancer Research Institute in 1953 specifically to preserve and advance her father's work. The toxins were still being used by some practitioners into the early 1960s, and that is when 1962 becomes impossible to ignore. In 1962, the FDA passed new regulations under the Kefauver-Harris Amendment that fundamentally changed how biological treatments were classified. Coley's toxins, which had been in clinical use for over 70 years, were reclassified as an investigational new drug. This meant they could no longer be administered to patients without going through the full FDA approval process from scratch. A treatment with 7 decades of documented clinical use was suddenly, legally, an untested new drug. Think about what that means. The approval process required controlled clinical trials. Controlled clinical trials required funding. Funding required a sponsor, typically a pharmaceutical company. But Coley's toxins were a mixture of killed bacteria. They could not be patented. No pharmaceutical company would spend hundreds of millions of dollars running trials on a product that anyone could manufacture. The regulatory reclassification did not ban Coley's toxins explicitly. It created an economic barrier that was functionally identical to a ban. Which brings me to what happened next. For the next several decades, immunology in cancer treatment was a backwater. A few researchers kept the thread alive. Lloyd Old at Memorial Sloan Kettering, ironically at the same institution that had marginalized Coley, continued studying immune responses to cancer. But the mainstream of oncology was radiation and chemotherapy. Billions of dollars flowed into drugs that poisoned cancer cells and healthy cells alike, hoping to kill the cancer faster than the patient. Meanwhile, Coley's core insight, that the immune system could be activated to recognize and destroy cancer, sat in published journals that anyone could access, but almost nobody read. Then in the 1990s, a researcher named James Allison discovered a mechanism called immune checkpoint inhibition. He found that cancer cells use specific proteins to hide from the immune system. If you block those proteins, the immune system can see the cancer and attack it. In 2011, the FDA approved the first checkpoint inhibitor, ipilimumab. In 2014, pembrolizumab, branded as Keytruda, was approved. In 2018, Allison won the Nobel Prize in Physiology or Medicine for the discovery. The Nobel Committee called it a landmark in the fight against cancer. And it is. But the principle, activating the immune system to attack tumors, was first demonstrated by William Coley in 1891, which brings me to the detail that does not add up. The official explanation is reasonable at first. Coley's toxins were inconsistent. Results varied between practitioners. The mechanism was not understood. When radiation and later chemotherapy offered more standardized, reproducible results, the medical establishment moved toward what could be measured and controlled. Science advanced. Old approaches were superseded. That is how progress works. I accepted that explanation until I looked at the timeline. But here is what that explanation leaves out. Coley's toxins were not abandoned because they were disproven. No published study demonstrated they did not work. They were abandoned because they were reclassified. And they were reclassified under a regulation that specifically disadvantaged treatments that could not be patented. The Eliava Institute in Georgia refined phage therapy over decades when early results were inconsistent. Western medicine could have done the same with Coley's toxins. The inconsistency was a solvable problem. But solving it required investment, and investment required a return, and a return required a patent, and you cannot patent killed bacteria. This was not a scientific judgment. This was an economic filter disguised as a regulatory one. The FDA did not evaluate Coley's toxins and reject them. They reclassified them into a category where evaluation became economically impossible. There is a difference between a treatment that fails a trial and a treatment that never gets a trial because no one can profit from running one. Coley's toxins are the second kind, and they are not alone. The timing is precise. Coley published results for 40 years. Ewing marginalized him for institutional reasons. The FDA reclassified his treatment for regulatory reasons. And 30 years after that reclassification, the same principal won a Nobel Prize and generated a drug that earns over $25 billion a year. The science was not wrong in 1891. The business model was wrong. There was no business model. And in American medicine, a treatment without a business model is a treatment without a future. I keep returning to Elizabeth Daschle, the 17-year-old girl whose death in 1890 sent William Coley searching through hospital records for an answer. He found one. It was not perfect. It was not fully understood. But it worked often enough, across enough patients, over enough decades, that his own daughter spent her entire adult life trying to make sure it was not forgotten. Think about what that means. Helen Coley Knotts did not spend 50 years defending a fraud. She spent 50 years defending documented clinical results that the system had decided were economically inconvenient. 70 years of clinical use. Over 900 documented cases. Published in the most respected journals in American medicine. And today, the average American has never heard of William Coley. But they have heard of Keytruda. They have heard of immunotherapy. They may even know someone who is paying $150,000 a year for a treatment built on a principle that a bone surgeon in New York demonstrated for the cost of a bacterial culture in 1891. Every time someone is prescribed a checkpoint inhibitor, they are paying for a principle that William Coley published for free. Every dollar of that $25 billion in annual Keytruda revenue rests on a scientific foundation that was laid by a man whose own hospital pushed him aside in favor of radium funded by a mining magnate. If this is the kind of investigation you want to see more of, subscribe. This is not the end. There are hundreds of these stories, and the pattern repeats with a precision that I cannot explain away. Maybe I am seeing patterns where none exist. Maybe Coley's toxins were genuinely too inconsistent to survive. Maybe radiation really was the better path, and the science simply needed 70 more years to circle back to immunology on its own terms. Maybe James Ewing was right, and the future belonged to institutions with expensive equipment, not surgeons with vials of bacteria. The simplest explanation is often correct. But then I look at Helen Coley Knotts, spending 50 years compiling her father's records because she knew what those records contained. I look at the 1962 reclassification that made it economically impossible to test a treatment that had been used on patients since 1891. I look at James Allison accepting the Nobel Prize in 2018 for a principle that William Coley demonstrated 127 years earlier. And I think about how many patients between 1962 and 2011 might have benefited from a treatment that existed but could not be legally administered because no one could patent it. 49 years. 49 years between the reclassification and the first approved checkpoint inhibitor. That is not a gap in the science, that is a gap in the economics. And gaps in economics are measured in lives. Tell me what you think. Am I seeing patterns, or is this what it looks like? The regulatory reclassification that erased Coley's toxins did not only affect one treatment. The same rule, the same year, the same mechanism was applied to every biological preparation that existed before 1962. What else vanished under that filing? That is its own investigation. And the list is longer than you think.